Welcome back to the deep dive. We're here again to take some really complex source material, break it down, and hopefully give you the tools you need to, get your head around some tricky topics.
That's the plan.
So if you've thought about genetics before, you probably know the basics: dominant recessive genes, maybe some specific single gene diseases pop into mind.
Like cystic fibrosis, perhaps, Huntington's, those classic examples.
Exactly. But today, we're diving into something different. It's actually the most common category of genetic conditions, but maybe the most confusing.
I think that's fair to say. Yeah, we're moving beyond that simple one gene equals one disease idea, the Mendelian disorders.
Okay.
Our mission today is really to tackle the complexity behind the diseases that honestly affect almost every family out there. We're talking about complex multigenic disorders.
Right, sometimes called multifactorial disorders.
That's the one, multifactorial. Like heart disease, type 2 diabetes, hypertension, those autoimmune conditions like lupus or rheumatoid arthritis.
the really big ones in terms of public health.
Huge. And the key idea is they're caused by many small genetic variations interacting with, well, everything else, your environment, your lifestyle.
So it's crucial to understand this distinction, right? These aren't the rare single gene problems or those big chromosomal abnormalities. These are much, much more common.
Massively more common. When you look at the overall frequency of all genetic diseases combined, it's these complex, multigenic ones that make up the vast majority of the disease burden.
So understanding them isn't just academic.
Not at all. It's fundamental for preventative medicine. It's the basis for where personalized medicine is heading.
Okay, so let's get into the mechanics then. What makes something like type 2 diabetes complex multigenic? What's the big difference at the DNA level compared to, say, sickle cell anemia?
The core shift, really, is moving from thinking about one big catastrophic mutation, something with a large, very obvious effect.
Like a major typo in the instruction manual.
Exactly. Instead, we're looking at multiple gene variations, maybe dozens or even hundreds, where each one individually only has a small effect, a tiny nudge.
So not one fatal punch, but maybe death by 1,000 paper cuts, genetically speaking.
That's a great analogy, actually. It's not an on-off switch. It's much more like a dimmer switch, or maybe a whole control panel of tiny dimmer switches adding up.
That sounds fundamentally different from the inheritance patterns we learn in basic biology.
It absolutely is. So the definition we use is that these complex multigenic disorders are conditions caused by interactions between multiple different variant forms of genes and environmental factors.
Okay, multiple gene variants in the environment.
Yes, and that's why the terminology is important. We say multigenic or polygenic, meaning many genes.
Poly meaning many. Got it.
And we also say multifactorial to really stress that environmental part. Lifestyle, diet, exposure, it's all mixed in. You really can't separate the two.
Let's stick with the genes for a second, though, those small nudges. In Mendelian diseases, the mutation is usually rare, often pretty damaging. What are these common subtle variations called in complex disorders?
Right, those are called polymorphisms.
Polymorphisms, okay.
Right. And the definition is really key here. A polymorphism is a variation in a gene where you find at least two different versions, or alleles, commonly in the population. Specifically, each version has to show up in at least 1% of people.
1%. So these aren't rare mutations, they're common variations in our DNA.
Exactly. That tells us they're not usually disastrous on their own. If they were, natural selection would likely weed them out pretty quickly. They're often neutral or maybe only slightly unhelpful in certain contexts.
That's a critical difference, common and low risk individually. So I could be walking around right now with, 20 different polymorphisms that slightly increase my risk for heart disease, but I'm perfectly healthy.
You absolutely could be. And this is where the concept of penetrance is so different from Mendelian disorder.
Penetrance. That's like
Yeah.
the likelihood that having the gene variation actually leads to the condition.
Precisely. For a classic Mendelian mutation, penetrance is usually high. If you inherit the faulty gene for Huntington's, for instance, your chance of developing the disease is extremely high.
Almost certain, right?
Very close to it, yes. But with these polymorphisms involved in complex diseases, each individual variant usually has low penetrance. It only nudges your risk up a tiny bit.
So using your analogy, a Mendelian gene is like carrying a bomb that's almost sure to go off. A polymorphism is more like carrying one slightly heavier book in your backpack. It adds a little strain, but doesn't stop you.
That's a really good way to put it. Now imagine you're not carrying one heavy book, but maybe 20 or 50 or even hundreds of slightly heavy books.
Okay, eventually that backpack's going to get pretty heavy. The strain adds up.
That's the summation effect. That's the core idea. No single polymorphism, no single susceptibility gene is usually necessary or sufficient on its own to cause the
It
disease.
has to be the combination.
Yes, the disease only really starts to manifest when an individual happens to inherit a specific, unfortunate combination of several of these low-penetrance polymorphisms. Enough of them stack up to push that person over some kind of physiological threshold.
And this whole idea, the stacking up of common variants, is basically the common disease, common variant hypothesis.
Exactly that. It's the prevailing model for understanding most complex disorders. It suggests that common diseases like type 2 diabetes or hypertension are primarily caused by inheriting a collection of many common low-risk gene variants.
So we're not looking for one big genetic mistake anymore.
Not usually, no. We're looking at the combined cumulative risk score from maybe thousands of these tiny genetic nudges. It's the collection that matters.
That completely changes how you think about hereditary risk, doesn't it? If a disease runs in the family, it's not necessarily one bad gene being passed down.
Right. It's more likely that family members are inheriting statistically similar collections of these common, slightly risky variants. But the specific combination will still be unique to each person, even siblings.
Which explains why these diseases often don't follow those neat Mendelian patterns on a family tree.
Precisely. The risk behaves more like a quantitative trait, like height. It exists on a spectrum. It's not just on or off.
Okay, so let's move from the concept to reality. Which specific diseases fall squarely into this complex multigenic category? You mentioned a few.
Yeah, the big ones that dominate healthcare today. The source material points specifically to things like atherosclerosis, hardening of the arteries, and essential hypertension, high blood pressure.
Okay.
Then there's diabetes mellitus, both type 1, which is autoimmune, and type 2, which is more metabolic, and also a whole range of autoimmune diseases. Think lupus, multiple sclerosis, rheumatoid arthritis.
And these are so common because the underlying polymorphisms, the risk factors, are themselves common in the population.
That's the thinking, yes. They're part of the normal spectrum of human genetic variation.
It's also really important, I think, to point out that this isn't just about diseases, right? This polygenic inheritance, this additive effect, governs lots of normal traits, too.
Oh, absolutely. The same genetic mechanisms, lots of genes, each contributing a little bit, are responsible for traits that show what we call continuous variation.
Continuous variation, like
Yeah.
things that exist on a scale, not in distinct boxes.
Exactly. Think about adult height. People aren't just tall or short. There's a whole smooth range, a bell curve, if you plot it for a large population. Same for body weight, skin pigmentation, eye color, even things like baseline intelligence quotients. These are all governed by hundreds of genes adding up.
So the system that creates disease risk is the same system that creates normal human variation.
Fundamentally, yes. It's just a question of whether the combined effect pushes you towards a state we define as disease.
Now, if we're talking about dozens or hundreds of genes contributing risk, does that mean they all contribute equally? Is every heavy book in your analogy the same weight?
No, not at all. And this is where the picture gets quite a bit more complex, actually. There can be significant variation in genetic significance even within a single complex disorder.
Meaning some gene variants carry more weight than others?
Much more in some cases. Certain genetic locations or loci can contribute a disproportionately large chunk of the risk compared to others.
Okay, can you give us an example?
Type 1 diabetes is probably the best studied example here. It's an autoimmune disease where the body attacks its own insulin-producing cells. Research suggests maybe 20 to 30 different gene regions are generally involved in increasing risk.
Okay, 20 to 30, that sounds pretty multigenic.
It is. But a small handful of gene variants within one specific region absolutely dominate the risk profile. The source highlights the HLA alleles.
HLA, human leukocyte antigen, that's related to the immune system, right?
Exactly. It's a complex of genes on chromosome 6 that essentially helps your immune system distinguish self from non-self. And certain variations, certain alleles, within this HLA region contribute, get this, more than 50% of the total genetic risk for type 1 diabetes.
Wow, over half the genetic risk from just one area.
It's a huge contribution. While there are definitely other genes involved, those other 20-plus variants that add smaller amounts of risk, this one HLA locus acts like a major gateway.
Wait a minute, though. If one spot contributes that much risk, over 50%, doesn't that start to blur the lines? Why isn't it just considered like an HLA-driven disease? Doesn't that sound almost Mendelian?
That's a really sharp question. It gets right to the heart of the classification challenge. We still call it multigenic because even that massive 50% plus contribution from HLA is not sufficient by itself to cause the disease.
Ah, okay. Necessary but not sufficient.
Kind of. Many people carry those high-risk HLA alleles and live their whole lives without ever developing type 1 diabetes. It still seems to require the additional contribution from those other lower-risk genes, the rest of the stack, plus the crucial environmental trigger.
So even with the main risk factor present, you still need the supporting cast of minor genes and the environmental push.
That seems to be the case. It sets a very high genetic predisposition, but you still need that complete picture, that convergence of genetic susceptibility and environmental factors for the autoimmune attack to actually happen.
Okay, that clarifies it. It's a major risk factor, but not deterministic on its own. Now, you also mentioned overlap between diseases. Do these polymorphisms pop up in multiple conditions?
They certainly do. It's rare for genetic pathways to be completely unique to one disease. If you look across different autoimmune diseases, for example, you'll often find shared polymorphisms.
Meaning the same gene variant might slightly increase risk for both, say, rheumatoid arthritis and lupus.
Exactly. These might be variants in genes involved in general immune system processes, like T-cell activation or pathways controlling inflammation. It tells researchers there are shared underlying mechanisms.
But then some variants must be specific.
Yes, alongside those shared risk factors, you'll also find polymorphisms that seem highly disease-specific. These might be in genes that influence which particular tissue the immune system targets, like the pancreatic beta cells in type 1 diabetes or the myelin sheath in multiple sclerosis.
Okay, so it's a mix of general and specific genetic risks. Given all this complexity, multiple genes, varying contributions, environmental factors, overlapping risks, how do researchers even begin to figure out if a disease truly belongs in this complex multigenic bucket, rather than just being a weird version of a single gene disorder?
It takes a lot of detective work. The first step is demonstrating clear familial clustering. The disease has to demonstrably run in families more often than expected by chance.
So it is inherited somehow.
Right. But then you have to systematically rule out the simpler explanations. Does it fit a classic Mendelian pattern, dominant recessive X-linked? No. Are there obvious chromosomal abnormalities involved? No.
So you look at the family tree, the pedigree, and the pattern just doesn't fit those neat boxes. It's kind of scattered, maybe skips generations unexpectedly, affects people with varying severity.
Exactly. The inheritance pattern looks irregular, maybe diffuse.
Yeah.
That's a big clue.
But you mentioned earlier there's a potential pitfall here, a gray area. What makes it hard sometimes to distinguish a truly complex disorder from a single gene disorder that's just behaving strangely?
The difficulty comes down to two concepts from Mendelian genetics: variable expressivity and reduced penetrance.
Okay, refresh my memory on those.
Sure. Variable expressivity means that even if everyone who inherits a specific single gene mutation gets the disease, the severity can vary wildly between individuals.
So same gene flaw, but one person gets a mild case, their sibling gets a severe case.
Precisely. And then there's reduced penetrance. That means not everyone who inherits the disease-causing mutation actually develops the disease at all. Maybe only 80% or 60% of people with the mutation ever show symptoms.
I see. So if a single gene disease has reduced penetrance, it might look like it's skipping generations or appearing randomly in a family, mimicking a complex pattern.
Exactly right. It can mask the underlying simple inheritance and make it look falsely complex on a pedigree chart.
So how do researchers tell the difference in those tricky cases? What's the giveaway for a truly multigenic condition?
Often, the biggest clue is the distribution of the trait or the disease risk across the entire population. Multigenic traits tend to show that continuous variation we talked about earlier, that bell curve distribution.
Like blood pressure.
Perfect example. Hypertension isn't just high or normal. Blood pressure exists on a continuous spectrum. You can plot everyone's blood pressure, and it forms a nice bell curve. That smooth, continuous distribution across the population is the classic signature of many small additive genetic effects, the hallmark of polygenic inheritance.
Okay, that makes sense. The population level pattern gives it away.
It's a very strong indicator, yes.
All right, so we've established the genetic side is complicated, involving many common variants stacking up, but the term is multifactorial. We absolutely have to dig into that other half, the environment.
We absolutely do, because this is where the concept gains so much clinical power. If the genes provide the underlying susceptibility, the environment is often what pulls the trigger.
So genetic risk might load the gun, but environment pulls the trigger.
That's a very common way to put it, yeah.
Yeah.
Environmental influences significantly modify how those genetic predispositions actually play out what we call the phenotypic expression.
The observable outcome.
Right. Genetic risk creates a vulnerability, maybe a predisposition, but it's often the environment, diet, lifestyle, infections, exposures that acts as the crucial modulator, determining whether that vulnerability stays hidden or flares up into actual disease.
Can we see this really clearly in specific examples, like the environment acting almost like a switch?
Oh, definitely.
Yeah.
Let's go back to type 2 diabetes. An individual might inherit a collection of polymorphisms that puts them at high genetic risk. They might have a high polygenic risk score, which we can talk about later,
Okay.
but they could remain perfectly healthy, metabolically speaking, for decades. Often the disease only manifests after a significant environmental shift, maybe substantial weight gain, a long period of eating poorly, becoming very sedentary.
So those lifestyle factors act as the key.
They seem to act as the key that unmasks the underlying genetic susceptibility. The environment didn't create the genetic risk, but it allowed that risk to become reality.
That really shifts the focus, doesn't it? It implies individuals have a lot of agency, even with a high genetic risk.
Immense agency. And it's not just for diseases. The source material gives great examples for normal traits, too. Think about height again. You can have identical twins, monozygotic twins.
Yeah, same exact genes.
Exactly the same genes, same genetic potential for height. But if one twin suffers from significant malnutrition during their critical growth years, while the other is well-nourished,
They'll end up different heights as adults.
noticeably different heights. The genetic blueprint was identical, but the environmental input nutrition in this case constrained the final physical outcome for one of them.
Or the example about intelligence. Someone could have all the genetic markers associated with high cognitive potential.
But if they grow up in an environment that's severely lacking in education, stimulation, social interaction, they simply won't be able to fully realize that innate genetic potential. The environment acts as a limiter, or conversely, an enabler.
So it's not just an on-off switch. The environment can kind of dial the expression up or down across a range.
That's a great way to think about it. Diet, stress levels, exposure to pollution or toxins, even infections. These are all inputs that interact with your baseline genetic settings.
The clinical implication here feels huge. If these gene-environment interactions are so central, it means we can potentially prevent or manage these diseases by focusing on the environmental side, even if we can't change the genes themselves.
That is absolutely the cornerstone of preventative medicine for most common chronic diseases. For those rare single gene disorders, the focus might be on gene therapy or replacing a missing protein. But for complex multigenic disorders, the power often lies in modifying the environment.
Managing weight, improving diet, increasing exercise, reducing stress.
Exactly. These become incredibly powerful tools. We're essentially trying to keep that underlying genetic susceptibility remasked to prevent it from manifesting, ideally indefinitely. It gives us actionable targets.
Okay, this brings us squarely into the modern era. We've got this picture of potentially hundreds of genes, each with a tiny effect, interacting with countless environmental factors. It sounds almost hopelessly complex. How on earth do researchers actually study this? How do they measure these tiny risks and figure out what's going on?
Yeah, it's a massive data challenge. It really required a technological revolution in genetics, which, thankfully, we've had over the last couple of decades. We needed tools to handle this sheer volume of information.
We moved into the era of big data in biology.
We absolutely did.
Yeah.
This is where personalized medicine starts to become a reality, leveraging technology to sift through signals across the entire genome.
And a key technology was one that allowed scanning the whole genome at once, right? Not just looking at one gene at a time.
Precisely. The development of genome-wide association studies, or GWAS, was a game changer. Starting around the mid-2000s, these studies really provided the evidence that confirmed the common disease, common variant hypothesis on a massive scale.
How did GWAS studies actually work? What are they looking for?
Essentially, you take huge groups of people, sometimes hundreds of thousands or even millions. Some have the disease you're interested in, say type 2 diabetes, and others form a healthy control group.
Okay, massive groups. Then what?
Then you scan their entire genomes, looking for millions of known common variations, those single nucleotide polymorphisms, or SNPs we talked about.
The polymorphisms, right?
And you're looking for tiny statistical differences. Is a particular SNP, let's call it SNPX, slightly more common in the group with type 2 diabetes compared to the healthy control group?
So it's purely statistical association.
Yes. It's a huge statistical undertaking. You're trying to find variants that might only increase risk by, say, 10%, a risk ratio of 1.1. That's a tiny effect.
Yeah.
But if you find that small difference consistently across 1 million people, The statistical association becomes incredibly strong. You can be confident that S&P is genuinely linked to the disease risk, even if its individual contribution is small.
And GWS studies found thousands of these associations for common diseases.
Thousands upon thousands. For conditions like heart disease, diabetes, schizophrenia, inflammatory bowel disease, GWOS uncovered hundreds, sometimes thousands, of SNPs scattered across the genome, each contributing a tiny piece to the overall risk puzzle. It painted exactly the picture the common disease, common variant hypothesis predicted.
Okay, so GWSA found the thousands of tiny risk factors. But how do you translate that mountain of data knowing someone has, say, 5,000 slightly risky SNPs into something practical for a patient or their doctor?
That's the next crucial step, moving from discovery to application. And that's led to the development of polygenic risk scores, or PRS.
Polygenic risk scores, PRS. Okay, what are those?
This is where computational power really comes in. A PRS basically takes all that information from GW, the specific risk associated with potentially thousands or even millions of common variants identified in those huge studies, and boils it down into a single score for one individual.
A single number summarizing my overall genetic predisposition for a specific disease?
Essentially, yes, based on your own DNA sequence data.
How is it calculated? Is it just adding up the number of risk variants I have?
It's a bit more sophisticated than just counting. It's usually a weighted sum. Each risk variant identified by GW is assigned a weight based on how strongly it was associated with the disease, how big its effect size was in the study.
So stronger risk variants contribute more to the score.
Exactly. The algorithm looks at your genome, sees which risk variants you have and whether you have 1 or 2 copies of each, multiplies the count by the statistical weight for each one, and then sums it all up. This gives you a single PRS number.
And what does that number tell me?
It places you on a spectrum of genetic risk relative to the wider population. Someone with a PRS in the top 1% for coronary artery disease might have a 3, 4, or even 5 times higher lifetime genetic risk compared to someone with an average score in the middle, 50%.
Wow. That sounds incredibly useful for prediction. What's the practical application right now?
It's becoming more and more integrated into clinical practice, especially in areas like cardiology and oncology. The idea is to identify individuals at high genetic risk before they show any signs of disease.
Proactive rather than reactive.
Exactly. If a patient comes back with a very high PRS for breast cancer or heart disease, even if they're young and currently healthy, their doctor can say, look, your genetic background puts you in a higher risk category. We need to be much more proactive with screening, lifestyle changes, maybe even preventative medication like statins, starting now.
That provides really actionable information for personalized prevention strategies. That's powerful. But thinking about the gene-environment interaction, is there any pushback or controversy around PRS? Does it risk making people feel like their fate is sealed by their genes?
That is definitely a major point of ongoing discussion and ethical consideration. A key criticism is that while a PRS might be excellent at quantifying genetic predisposition, it doesn't inherently account for the environmental side of the equation.
Right. The score tells you about the loaded gun, but not whether the trigger will ever be pulled.
Precisely. Someone could have a high PRS but maintain an exceptionally healthy lifestyle and potentially never develop the disease. Conversely, someone with a low PRS could still develop the disease due to very strong negative environmental factors. The PRS is a powerful piece of the puzzle, but it's not the whole picture.
So it tells us about the shaky foundation, maybe how shaky it is, but not necessarily about how well you've reinforced it with lifestyle choices.
That's a good way to put it. It quantifies the innate susceptibility, but the manifestation still depends heavily on that environmental interplay. And this brings us back to the deeper biological question. How does the environment actually talk to our genes? What are the molecular mechanisms?
Right, we talked about lifestyle unmasking the genetic risk. How does that happen at the cellular level? What's the machinery?
This leads us right to the cutting edge of research, into epigenetics and gene regulation.
Epigenetics, that's changes in gene activity without changing the DNA sequence itself.
Exactly. Think of it like adding sticky notes or volume controls to your DNA instruction manual. The underlying text, the DNA sequence, including all those polymorphisms, doesn't change. But epigenetics determines which genes are read loudly, which are read softly, and which are skipped over entirely.
So the environment isn't rewriting the genetic code. It's controlling how the existing code is used.
That's the core idea. And the mechanisms are things like DNA methylation, chemical tags, methyl groups, get attached directly to the DNA, often acting like off switches, silencing gene expression in that region.
Okay, methylation is one mechanism.
Another major one is histone modification. Histones are the spool-like proteins that DNA wraps around to fit inside the cell nucleus. Chemical modifications to these histone proteins can either tighten the DNA winding, making genes inaccessible, or loosen it, making them easier to read and transcribe.
So environmental factors like diet or stress can actually trigger these epigenetic changes. How does that connect back to the polymorphisms?
Well, imagine you have a set of polymorphisms that slightly increase your baseline inflammatory response. Now, let's say you experience chronic stress or consistently eat a pro-inflammatory diet. Those environmental signals can trigger signaling pathways inside your cells. Those pathways can then activate the enzymes that add or remove epigenetic marks, the DNA methylation or histone modifications. These marks might then land near your inflammation-related polymorphisms, potentially turning up the expression of those already slightly risky genes.
So the environment signals the epigenetic machinery to amplify the effect of the underlying genetic risk variants.
That's a key part of the model, yes. It pushes the system closer to, or maybe over, that physiological threshold where disease symptoms start to appear. The environment provides the instructions, and the epigenome executes the changes on your specific genetic background.
And the source material also mentions non-coding RNAs being involved in this regulation.
Yes, absolutely. That's another layer of complexity and control. For a long time, we focused mostly on genes that code for proteins, but it turns out vast stretches of our genome produce RNAs that don't make proteins. These are the non-coding RNA.
Like microRNAs.
Exactly. MicroRNAs, miRNAs, and also long non-coding RNAs, LNCRNAs, are key players. They act like regulatory molecules. miRNAs, for instance, can bind to messenger RNAs, the templates for making proteins, and either block them from being translated or mark them for destruction. They're like fine-tuners of gene expression.
So environmental signals could also change the levels of these regulatory RNAs, which then affects how the protein coding genes, including those risk polymorphisms, are ultimately expressed.
Precisely. It's this incredibly intricate dynamic network. Environmental inputs influence epigenetic marks and non-coding RNAs, which together modulate how your specific set of inherited polymorphisms actually functions from moment to moment. Every meal, every stressful encounter, potentially tweaks this complex regulatory system.
Wow. Okay, this has been a truly deep dive, moving from basic definitions all the way to epigenetics. If we try to synthesize all of this, what's the main takeaway message for someone trying to understand these common complex diseases?
I think the core message is really about scale and control. First, these complex multigenic disorders are by far the most common type of genetic condition. They arise from the combined additive effect of many common, individually low-risk gene variants. It's not about one big flaw. It's about the overall stability of the genetic foundation.
A shaky foundation, as we
It's
said.
a potentially shaky foundation, yes. But, and this is the crucial part, how that foundation actually performs, whether it leads to problems, is heavily, heavily influenced by environment and lifestyle.
And that interaction, that environmental influence, is actually where our power lies.
Absolutely. Unlike some rare single-gene disorders that can feel almost deterministic, the multigenic nature of common diseases highlights the enormous leverage we have through environmental modification.
So genetic risk is definitely not genetic destiny here.
Not at all. In fact, the more we learn about the specific genes and pathways involved through things like GWS and PRS, the better we can potentially identify the most effective environmental interventions. The specific dietary changes, exercise routines, or stress management techniques that can counteract an individual's specific genetic risk profile and help keep that susceptibility masked.
Which brings us perfectly to our final provocative thought for you, the listener, to consider. We talked a lot about how negative environmental factors like weight gain or inactivity can unmask a hidden genetic risk for something like type 2 diabetes.
Right.
So let's flip that. Thinking about the future. What are the specific, scientifically validated environmental factors, nutrients, exercise protocols, maybe even psychological practices that could be strategically used to intentionally remask or actively mitigate the risk conferred by the strongest known polymorphisms for common diseases? Could we tailor interventions to counteract, say, the high risk from specific HLA alleles in autoimmune disease, or the top SNPs for heart disease identified by GWSs?
It really pushes us to think about personalized prevention at a molecular level. What are the precise environmental levers we can pull, based on solid science, to most effectively shore up our unique genetic weak points? That's the frontier.
A fascinating challenge and a hopeful note to end on. Your genetic blueprint might be set, but the journey you take with it offers so many possibilities for intervention. Thank you for exploring that complexity with us today.